Welcome back to your Unit 3 study guide. We're starting on page 3. The other video had page 1 and 2. In page 3, we pick up with number 5, where it says to match the following organelles to their functions. The first one here is going to be the cell wall is letter B. It provides structural support for the cell in plants only. So strong, rigid structure for plant cells. Letter F was cell membrane because while the cell membrane provides structure, it also controls what enters and leaves the cell. H is the cytoplasm. The cytoplasm is the jelly-like substance inside of the cell. It's kind of like the water inside of a water balloon. It supports organelles. It gives them something to kind of float around in. And it's also where your chemical reactions happen. You need to have a solution, an aqueous solution, for the chemical reactions to happen inside of your cells. Letter C is the nucleus, the control center, the brain of the cell where the DNA is found. D is the mitochondria. This is the mighty mitochondria that's going to produce energy from food or whatever type of substances the cell uses for energy. J. Is that a J? Yes. J is a chloroplast. It is the site of photosynthesis, and it is the place where cells are going to create food from sunlight, water, and carbon dioxide. So that's photosynthesis. E is a vacuole. A vacuole is found mostly in plant cells that stores food, water, or waste. It's a large central vacuole. A is the endoplasmic reticulum. It's a transportation network that picks materials up as they leave the nucleus. So it'll take information that's coming out of the nucleus and kind of transport it through the endoplasmic reticulum so it can create proteins and then get those proteins to where they need to be. So the ER is the transportation network. Golgi apparatus is the post office. It packages materials for shipments either within or outside of the cell. A vesicle is a membrane-bound structure that carries materials into or out of the cells, and vesicles are typically made in the Golgi apparatus. And finally, a ribosome is the site of protein synthesis. This is where proteins are made. If you want a little bit more details about organelles that have pictures, go back to the organelle video that I made for you when I showed you the answer key for the plants versus animals lab that we did with the microscopes. So you can review that video as well, just to kind of review your organelles with pictures. Number 6 on page 3 is talking about active transport versus passive transport, which we did a few times. You have passive transport, which is diffusion and facilitated diffusion, also osmosis. This does not require energy from the cell. Active transport is endocytosis and exocytosis. This does require energy from the cell. Please review the transport videos that I've posted on Schoology because I do go into a lot more detail with that there, and I also have some pictures. So that's another resource for you to use to study from. I'm going to flip over to page 4. We are almost done. This is something that they like to do on the Keystone exam, so we included it on the study guide, but they'll like to give you a really long, wordy question that has a pretty simple answer to it. An organism's ability to maintain a stable internal environment while there's a change to the external environment is known as homeostasis. How do our cells maintain homeostasis in response to the environment around them? There is a Crash Course Biology video in the materials section of Schoology if you'd like to watch it. It's called In the Club. It's with Hank Green. But it's interesting. He talks about homeostasis in the cell membrane. So the cell membrane is selectively permeable, controlling the substances that enter or leave the cell. It can deny some substances. It can dump stuff out if we need to reduce the concentration. It can allow more water to come in. All of those things that we talked about with transport are how the cell maintains homeostasis. We observed our gummy bears respond to the environment by allowing water to go inside and outside of it, just like a cell does. Describe how the gummy bears changed in response to their surroundings in terms of them being hypotonic, hypertonic, or isotonic. So in the hypotonic solution, there were fewer solutes on the outside, which was going to cause the gummy bear to become hypertonic, and he would expand or burst. In a hypertonic solution, there's more solutes on the outside, and this is going to cause the gummy bear to try to expel some of its water. Now, most of our gummy bears in our hypertonic solution just kind of turned into mush. They just melted because a gummy bear is different from a cell. It's not equipped to have water leave the gummy bear. And then isotonic is just right. You've got the same or an even concentration of solutes on the inside of the cell that you do on the outside of the cell. So same. All right, this concludes the Unit 3 study guide. If you have any questions about anything, please email me before the test. And if not, good luck tomorrow.